Welcome everyone. This is our third presentation, live webinar with Motic and Media Cybernetics. Previously, we've done a live cell imaging. We've done also an industrial inspection and grains analysis. And today we're moving into the food and beverage R&D and quality control. A lot of, I think we have joining us today, a lot of our partners, as well as some microscope users in the food and beverage industry. Today we'll cover off both the microscopy applications most recommended for certain food and beverage applications, and our Media Cybernetics partners will cover off the key software applications used, both regular analysis applications and some of their AI applications. I'm Alex Lobazar, been with Motic for almost 20 years, commercial director. And I also have Keith Ledford, who's our national sales manager, joining. He'll be covering off the microscope sections. And then we'll have Nick Beavers and Tom Moyer from Media Cy cover off their software expertise. Just to give you a little bit of a rundown on the Motic side, in case many of you don't know, we're a global manufacturer of optical instrumentation. We cover multiple markets: education, research, industrial, and not only offer microscopes, but offer digital cameras, offer whole slide scanners, usually for high quality and most often high quality observation for scientific discovery. So we are probably number 5 in the world as far as manufacturing size. We've got over 1,300 employees, 5,000 institutions are using our instruments. We're selling approximately 50,000 microscopes a year, and gives you an idea of our global footprint. Offices in Germany, Spain, China, Hong Kong, and I'm presenting now out of our Canada office. We have our Motic Swift line also, and our warehouse out of San Antonio, and our digital pathology office out of San Francisco. So we'll cover just a little bit of the introduction between Motic and Media Cybernetics. Motic's long been a significant player in the optics market. A couple of years ago, it came to our attention that, the image was a means to analyze, but Motic really didn't have the software, I would say, capability to provide advanced image analysis. And so we had selected we feel is one of the best partners in the industry and media side to join us and collaborate on a number of different application-specific software bundles. So we've gotten now the general bio, which are most of the software and hardware instrumentation combinations today you'll see is on the general bio side. But we also have a clinical combination, we do a research combination, and also a material science combination. So any of the products that you see today, or if there's any other industries you're interested in marrying up a microscope and or software application, we're open to discuss further any one of the industries that we currently cover. But let's get down a little bit into our topic of the day. We're talking about the food and beverage microscopy industry. So just to give you sort of a rough down, I'll cover the overall sort of, when I was taught microscopy earlier, it's always the magnification levels, and the type of microscopes that, are used in specific applications. So really, if we narrow down into the food and beverage area, we want to figure that the microscope type will explain the magnification and then sort of the usages. So bright field compound is, I would say, as proven here, is the highest, most commonly used type of microscope that also gives you your highest magnification. So you're looking at 40 times up to 1,000 times magnification. This is your conventional scope. You're looking at a glass slide, or you're putting your sample on a glass slide and putting a cover slip over top of it. And the primary applications for this are bacteria, yeast, molds, and food particulates. Then you get into sort of a lower magnification range, which is your stereo microscope. This traditionally is, say, a 5 to an 80 times or 5 to 100 times magnification. Usually about 20 to 25% of the market uses this type of stereo for basic inspection. So if you're looking at foreign material inspection, insects, fibers, packaging defects. This pretty much falls under sort of a stereo microscope look. Usually these types of samples are solid in nature, where light is not enabled to pass through it, so you're not really going to use a bright field scope. Then you get into some of the specific applications of light usage and microscopy usage. The main one really is phase contrast. So you'll see that's also similar to it's a bright field derivative with specific optic and optical additions. So that's a 100 to 1,000 magnifications. Here you're looking at live microorganisms, yeasts, bacteria, usually something without a conventional type stain or shade. The other contrasting method is a polarized light application. Again, that's used on mostly an upright microscope, and that's 40 times to about 400 times. And then you're looking at starches or sugars or fat crystals and adulterations. And then not only, I would say, in the food and beverage industry, but also in a lot of the other I would say life science, biomedical, and pharma industries, you're seeing more and more usage with fluorescence microscopy. That's becoming more, I would say, more popular, more engaging. Then you're still with an upright scope. You're looking at 100 times magnification to 1,000, and you're looking at, say, microbial detection or biofilms or specific pathogens. Just to give you an idea, if we're out searching, hunting for different industries that would benefit from microscopy applications, won't get into each of the applications here. We're happy to share the PowerPoint with you should you want to get a copy of it. Just let us know at the end. But the major industries where microscopy is used is dairy, brewing, or the brewery industry, looking at a lot of the yeast capabilities. I've noticed an explosion in that, especially in the craft beer market over the last couple of years. The wine industry, the meats industry, parasites, muscle structures, pathogens, specifically seafood applications, bakery applications, manufacturing of chocolate, say coffee or teas, bottled water, particulates or microorganisms, and then the soft drinks, yeast, mold, production contamination. So these are some of the industries, main industries, I would say, that benefit from a microscopy and software solution. Now I'll pass over to Keith. Keith will get into some of the individual microscopes and breakdowns of each of the different groupings. Keith?
Thank you, Alex. Hello, everyone. Thanks for joining us today. We're just going to give you a brief overview of some of the actual equipment, the microscopes that will be used for this. Of course, we're going to focus mostly on the image software with Image Pro, but just to give you some basic ideas, we talked about the common types of microscopes and the area applications they cover. In this case, the most common is our bright field compound microscope. Here you can see an example of our PA43 Bio and our Panthera C2. Those are equipped with our S6 camera, and you can see some imagery here from our Image Pro package. We briefly discussed stereo microscopes. Stereo microscopes are very common in the application where we're doing inspection and looking for contaminants and those type things. In this case, you're seeing we cover stereoscopes from the basic all the way up through research. Two of our most common ones are the SM7, which is our sort of entry-level research grade stereoscope, and our most popular model, the SMZ172. It's actually brand new, but is a continuation of our SMZ171, which has been our most popular model for several years now. Very capable and very affordable. As Alex mentioned, different contrast methods are used. Phase contrast is a common method used for some of the live yeast, live bacteria, and other general industries like beer, wine, and your yogurts and cheeses. In this case, we can add phase contrast to both our inverted scopes and our upright scopes. We talked first about our upright compounds, and the one to the left is our AE31E, which is an inverted scope, which is typical for live cell operation. Both of these are capable of phase contrast as one of the ways to image our live cells, live bacteria. Continuing on with the different method for contrasting images is polarized light. Polarized light is often used when we have crystals. You can see that to the left, some of the applications. In this case, there's a polarized light application with potato starch. That also is a popular addition to some upright scopes, and we have dedicated polarized scopes as the Panthera Tech Pole you see here. The Panthera Tech Pole offers quite a bit of capability with the rotating stage, rotating Bertrand lens, and the items you need for polarized microscopy. And lastly, on the microscope list, although it's a smaller part of this, as we grow and things progress, I think as Alex mentioned, we're getting more and more calls in different areas for fluorescence microscopy. So in fluorescence microscopy, we can offer you several packages, both in inverted and in uprights. Here you see our PA53 and FS6, and that is with our Image Pro software. You see an image in the background. This is great for several applications you see listed on the left, and the PA53 is a top of the line research level fluorescence scope, which will give you tons of capability built in right there, including your cubes on a turret and very nice detection on those. As far as the illumination, we have several features in the upright analysis scopes like this that give it more modern feel and all the bells and whistles you could need for something like this. The fluorescence can be added to some of our other scopes. We have some limited fluorescence applications on our Panthera, but we also have fluorescence applications for inverteds if needed. So that covers the majority of those for that part of it.
All right, just before we break into the media side, we wanted to just section it off a bit. Any quick questions on any of the microscope topics that we went over?
Please feel free to raise your hand or list that in the comments. I think May put a list out there, and we'd be happy to answer anything you have at the end as well if you want to just type it up.
Okay, looks like we're clean. I'll pass you over to Nick Beavers. He's the president of Media Cybernetics. Nick?
Thank you, Alex. Appreciate it. I really appreciate Motic joining us today on this webinar. What Tom and I are excited to talk to you about is a lot of different opportunities that are available to you with the packages that Alex and Keith mentioned. One of the interesting things about the Image Pro software that we're going to demonstrate to you live today is the ability to work with products that you may be facing every day on two different sides, maybe of your business. So whether it's micromaterials and ingredients or it's manufactured end products, what we're going to talk about today is how the Media Cybernetics Image Pro software is going to be able to handle images that you're capturing on your Motic microscope or other microscopes that you may have in your lab, and how we're going to work with them all using one software package. So on the left-hand side, obviously you're going to have a lot of properties of your micro materials and ingredients that could be very different from what you're going to see in a manufactured end product, and you have different challenges you're going to face as well. So on the next slide, let's just talk about, with these two different ways, what are some of the things you're going to see in your micromaterials? Well, first of all, you're probably going to see a lot of granule size and shape. You're going to want to understand particle size distribution. You're going to maybe want to look at different types of crystals, different types of ice structures. You're going to want to look at cellulose, protein strands, oils, emulsifiers, and maybe even yeast and bacteria. And all these things are going to come from, obviously a wide variety of micromaterials and ingredients. All these things are going to have particular properties that are going to require, one, a different microscope at a higher magnification, most likely, but also something I'll talk about in a later slide, maybe some interest up front in thinking about how you're going to approach the entire microscopy and imaging problem before you really get started. So the next one, on the next slide, we're talking about manufactured end products. And this, again, you have a different look and feel. So maybe you're looking at cellular or foam structures in baked goods. You're not going to see something exactly like this in your micromaterials. But the point I want to make is we're going to demonstrate today how just because it is a different image, and it is a different look and feel altogether of what you've captured. There is a lot of similarities because a piece of software is looking at this image as pixels and it's looking at intensities. And so regardless of the magnification, regardless of the color, it's going to be treated in a very similar way. So you may see things such as, films or gel fiber networks, cracks. You may see inconsistencies or mixed particulates. Regardless, again, of what you're seeing and what the color is and how it's distributed, you may actually get a very interesting revelation today about how we can approach this. And on the next slide, I want to talk about really what is the first step. So step one is going to be understanding, based upon all these different types of materials and ingredients you may be working with, what is my end goal? So I want to try to establish whether it is a physical structure, what it is, so some kind of classification of the biological content, let's say, what is a distribution of different materials, or what is there that shouldn't be there, such as some kind of defect or contamination. And just setting that expectation up front is going to help you with the workflow of your analysis. Now, I'm excited to set up Tom today. Tom's got a really great live demo. And what I'm excited to tell you about is something called Image Pro AI. And so in Image Pro 11.1, we introduced artificial intelligence segmentation into the software. What is, Tom and I have both been doing this for quite a while, and over a couple of decades, I have been so used to threshold-based segmentation, or kind of an older version of machine learning. And if you hear the word machine learning, it's a really impressive capability, and it was the best we had for a number of years. Put aside whatever you may think about AI and all the other aspects of life, but we can definitely say that it's in the microscopy and imaging world. It is an absolute game changer, and it has introduced a capability you didn't have for the entirety of microscopy and imaging. And it really introduces an opportunity to do things with these types of, complex structural challenges, or complex distribution challenges, contaminations that were really hard to pick out. AI really changes a lot of things. And so step one, it is important to understand what you're looking for, because it will help you to decide whether or not you're going to use different types of segmentation methods or manual methods of doing your analysis. But it's also going to set up how you should capture your image. And so how your microscope sees an image and sees the materials under your objective lenses is going to be a little bit different. So I just wanted to point out, as step number two, obviously you want to know what you're trying to get, but then how am I going to actually get it in my image? So with your naked eye, maybe you can see this, maybe you can't. What we talked about earlier, and Alex pointed out, is that there's a lot of different techniques, and the Motic microscopes offer all of these different techniques: bright field, dark field, polarized, phase contrast, fluorescence, and stereo. But why would I use these? So you're looking for general morphology when it comes to the analysis. Bright field's great. You get a nice, beautiful image. You can try to understand outlines, general morphology of whatever structure it may be. But what it might miss is something like edges. So if you had transparent particles, such as like a sugar crystal, you might want to try a dark field to be able to get those edges to really pop, if it's really important for you to identify those, because maybe it's not possible in bright field. Now, AI may change that a little bit. What we're seeing with AI is really, really dim images. Could be possible. And so what it might be useful is sometimes trying different microscopy techniques, if you have that available to you, for a single image, to be able to try to use it in analysis to get the best possible result. Polarized light is great for patterns. Phase contrast, you'll be fantastic for low contrast and transparent things. And of course, fluorescence can be used not just for fluorescent dyes or stains, but sometimes it can be really good at pulling out natural fluorescence and natural tracers, or some kind of tracer that may be put in there. And then lastly, of course, texture-based analysis is really going to be much more easily identified with some type of reflected light or even stereo-based technique. So keep those in mind as number two, I want whatever is in my image to have as best contrast as possible. I want it to be easier to be able to find what it is I'm looking at, even just with the naked eye, but I'm also trying to make that easier for the software as well. And so I have established what I want. I've chosen my method of microscopy. I've captured my images. And of course, the last thing, kind of teeing up Tom to give you this great demo, if I open up Image Pro, I need to be able to capture certain types of measurements. And what we're going to offer you today is a variety of ways of capturing literally thousands of measurements. Obviously, you don't probably need thousands of measurements, and that's why we produce these protocols that allow you to do a very simple particle count and sizing, and it does just that. Or something that's a particle morphology, just that. Color and texture analysis, defect identification, or area and coverage. And the kind of measurements on the right-hand side, if it's important for you, distribution curves, roundness, aspect ratio, understanding some kind of scoring mechanism, classification of different items, or voids and percent areas and distribution, all that's made possible. And a lot of the times what it starts with is either one of two things, either a manual measurement that's going to be obviously more time consuming. You see that in a lot of software. But what we're going to demonstrate today is a time savings where you have an efficiency that you can apply repeatably that can be across users, will be more consistent regardless of who's using it. And that's a concept called segmentation. And segmentation is something that we're probably going to really dig into a lot today and talk about how that can be integrated into protocols to make this a lot easier for you. So we have our 3-step process. And with that, why don't we talk a little bit about analysis? And for that, I'll hand it over to Tom.
All right. Thank you, Nick. All right, so before I get into the analysis portion, I just want to mention a little bit about the combination of Motic hardware with our Image Pro software. And one of the biggest benefits here is that you can acquire an end-to-end workflow. Again, the food industry, there are so many applications. It's such a broad range. So Motic has got equipment for just about every application, and I will demonstrate how Image Pro can tackle any of these applications as well using the same exact process. So if you combine the Motic microscope and camera with Image Pro, you will have the ability to take advantage of capture features. We do support every Motic camera in the S series line. So all of these packages that are pre-configured have the S series camera. So you'll be able to capture images directly into Image Pro. You won't have to go in between any sort of acquisition software and analysis software. So here you can see I have an image that I've captured from my SM7 stereo microscope. But then we also have some processing features as well. So I have added vignetting to this image. Let's assume that for some reason, whether it's lighting or something else, you're getting vignetting or some sort of uneven background. We can also do color corrections as well. So we have many processing features. All of this is done before you get into the analysis workflow. Then, of course, the main part of Image Pro is going to be the analysis workflow. So that starts with measurement, and that would be finding objects or areas, patterns within an image, maybe classifying them. And finally, reporting. Our goal is always to set up some sort of workflow where you can click a single button and you can just batch process as many images as you like without having to go through that step-by-step workflow every time you want to perform the analysis. So I just want to highlight a few capture features. These are a few features that are unique with the capabilities within Image Pro. And I'll go through this very quickly, but live high dynamic range. So you can capture several images of different exposure times, and that will eliminate anything. If you have a reflective material, this is really a really good feature for that. We also have live extended depth of field. So if you are using a stereo microscope and you need to capture the bottom of your sample and the top all in focus, we allow you to perform live EDF as well as live tiling. So you can move your manual stage around and Image Pro will automatically stitch the tiles together and create this nice mosaic image. We do have live measurements, so you can perform live manual measurements on your image. live grids. You can do a comparison between a captured image and a live image, as well as time-lapse. Two processing features that I want to highlight. One, again, would be shading or background correction. So whether this is lighting or hardware, we have the ability to correct backgrounds, bright and dark, and this can be based on another background image that you have, or you can set a background from different points. But a very easy process, and then with that, you can batch process and perform that operation, again, on a large number of images. I know within the food industry, if you are interested in extracting any sort of color information from your images, this one's actually extremely important. We allow you to do a color correction or transformation. So if you have some sort of known color value, this could be a color palette, or if you do have a color profile, we allow you to capture an image, and then we have a tool where you can look at the color palette and do a color transformation. And again, you can then apply that to an entire batch of images. So getting into measure and report, this is really our focus for today. We want to capture the images, bring them into Image Pro, but then we want to perform analysis. So when it comes to measure and report, I have 4 different samples and application case studies that I want to go through today, starting with fat globules and distribution of fat globules. Then we'll take a look at seed classification and color analysis. We have cereal morphology and structure, and we'll end with chocolate coating composition, so getting percent area measurements and ratios of different coatings on pretzels. So again, we follow this same 4-step analysis workflow for just about every application. And it's not just every application within the food industry. This is any application that you could think of, life science or materials. And it's very rare that we have to step outside of these 4 workflow steps. Number 1 is start with a pre-trained AI model. So Image Pro does come with a library of pre-trained models, so you can start right away. There's no need to immediately train a model to detect your objects. A lot of times we can start with a pre-trained model and it finds a very large percentage of objects accurately. If needed, you can easily clone and fine-tune that model. And then we move into the automation portion. And we do this by way of what we call protocols. These are automated workflow templates where you walk through them one time, you configure them, point to some models that you'd like to use for segmentation, add any additional measurement types that you want output at the end. But again, that's one time. You go through the workflow, set it up, then you can save it and batch process images. And that is step number 4, running automation after you have configured the protocol. But before we start, Nick mentioned other segmentation options. So if you are currently using or have tried to use analysis software in the past, chances are you have used one of these 2 segmentation methods. The first being the most basic. This is threshold segmentation, where we're trying to separate objects from the background, and this is all based on an intensity range. And I chose the globule application up front because even though visually I can see where these globules are, when it comes to image analysis, it's actually a very difficult thing to extract the globules from the background. And that's just because we have a dark outer rim, but then the inside of the globules shares a lot of the same intensity values as the background. So if I just go off of intensity values, you can see here I'm picking up a lot of the background, and if you look on the right side of the image, I'm not even picking up any of the globules. So this is not an extremely accurate method, really only if you have very high contrast objects, let's say dark objects on a white background, and they're not touching. And then we have machine learning segmentation. We call this smart segmentation. This takes into account a couple different characteristics, some color, texture. It does create a more robust segmentation. However, it struggles with some of the same pitfalls as threshold segmentation. So if I set up this machine learning, you can see in general, it's kind of ignored the background in most spots, but it's creating this halo effect around a lot of the globules. So again, it is a little bit more accurate, but it is far from repeatable for most applications. So step one, let's watch a video here. I'm going to show how we can apply an AI deep learning model by going into the library and choosing one of the pre-trained models. So this is my favorite model. It's called General Objects. This one can be applied to just about any image to find blob-like objects. And I use blob-like, that term very loosely. You can find, we actually use it to find fibers, so even wide aspect ratio objects. So all I do is open it up and I click on count, and I can see how well the model performs without doing anything. I just applied the model. And you can see here, it's actually done a really good job without any training of finding these globules. So if you move around, you can see it's also doing a good job of splitting the globules, which the other segmentation methods, you have to perform another function after the segmentation to split. And again, that comes with its own challenges. However, it has missed a few of the large globules, and that's what's most obvious when I'm zoomed out. But when you zoom in, it's also missed some of the smaller globules. So if that's the case, all we have to do is clone and fine-tune the model. We just have to give it a little bit of additional object references. We can delete things that it found that we don't want. We can add things that it's missed. So I'll walk through that process now. So after we run the prediction, we have a button called Clone Model. So it's going to copy the model. I can give this a new model name, which I'll call Fat Globule Model. And then I simply have to load it in our training panel. Now, again, the idea here is to label things that it's missed or to delete things that you don't want it to find in the future. And it would actually take quite a long time. It's deceptive if you're not zoomed in. But if I had to label everything within this image, it would actually take quite a long time to do. So we give you the ability to draw this region of interest. If I back the video up a little bit. A lot of the time, it just takes a single image, and not even the entire image, but just a small space within a region of interest. I just go ahead and label things correctly within that small square. I go ahead and train the model, and now I have something that is extremely accurate. Now, you could have to go back and give it some additional training by adding more reference labels, but I found that with most applications, you will see a vast improvement just using this small, tiny region within the image for training. So in the training panel, we have some tools, whatever your preference may be. We have a brush tool that allows you to circle objects. We also have polygon, square, circle, ellipse. Really, this is up to you. Some tools are better at labeling certain structures than others. But the idea is just color in the objects the way that you want the model to perform after it's done training. So this video is now being sped up because this would take quite a while. Even at the small ROI, it's quite a few labels. It's missed a few of these smaller globules. Once you have everything within that region of interest labeled, I can just add these new labels in as references and then click on train. And this is a quick process. It takes about 20 seconds. And now I have a model I can test out, and I can see if, with the training I've provided, if it's made any sort of improvement. So here we're training. So this is about a 20-second process, and we have graphs that show you, basically give you a visual indication if the model is training correctly or not. You don't want to see the graph going up and down and up and down. But we will save the AI technical side either for questions or for another webinar. Now that I've trained the model, I can open it back up in that prediction panel. Let's go ahead and click count, and we can see if it's made any improvements. And you can see it found the larger globules. It's also found some of the smaller ones as well. So this is really based on what you are interested in finding in your image. If you want to ignore some of the really small globules, if you train the model by not labeling those, then when you go to do the prediction, it's not going to find them. If you want those included in your segmentation, when you train the model, just circle those smaller globules, and then when you run the prediction, it will find them. So that is the segmentation process using AI. Again, we use the same training tools, prediction tools, for all of these applications. This could be training a model to find bread, either the whole piece of bread. It could be the voids within. You have to learn just one set of tools, and now you can apply that to any image, any application. After we have the segmentations set up, now we want to move into creating some sort of workflow. We found the particles, or we found the objects of interest, but now we want to create some sort of workflow that's going to maybe classify those particles and then automatically output certain types of data. So in step number 3, this is where we're going to walk through one of the pre-configured protocols and just make any changes that we need to before we save it for future use in terms of batch processing. So I'm going to go into the protocol library, and much like AI deep learning, we have libraries for essential protocols. These are sort of basic protocols for object count, objects within objects. We have a materials and a life science collection, but these are just templates. So you are not restricted to the steps and features within the protocol. We just try to create common workflows. That way you have a starting point, but you can fully customize these and add additional steps, etc. So in step number 1, I just need to label my classes. And since I only have a single class here, I'm going to call it globules. And then I get a visual indication of how many steps I need to perform to set this up. So step number 1 is setup. So that's already done. In step number 2, it's prompting me to pick an AI model that I'd like to use for segmentation. So I can use one of the default pre-trained models, but since we've trained one to find globules, I'm going to use that. So it's found all of my globules, and now in this panel, I have a couple different options. The first option is some sort of measurement range or threshold. Now, this historically was used a lot with threshold and machine learning based segmentation, because those segmentation methods weren't very good at not picking up background and not finding things that you weren't interested in. However, if you do have tolerances that you need to stay within, of course you would train the model to not find those objects, but if the model were to pick them up, then setting these, let me go back a little bit, setting these ranges is going to allow you to have actual values where you can exclude and ignore objects that don't fit within those ranges. And just to show you what you have the ability to output in terms of measurements, this is showcasing our measurement library. everything from area, region information to color values, texture-based measurements. Chances are, if you're looking to extract some sort of measurement, it's already in the library. However, you can also add your own custom formulas and expressions. So I've just added roundness into what I'd like to collect when I run the analysis. And now I have the ability to set a range based on roundness. So you can add as many different measurement types as you'd like. That can be for ranges, classification, or for data output. Now, also, we have an option for classification. So we can classify by a single measurement type, which I'm doing here. I can create as many different bins or ranges as I'd like. And I can spread the ranges out evenly. I can perform a log on those ranges, or I can manually enter known ranges. I can give these class names, etc. I can also change how I'm classifying objects before I move on to the next step. So here I'm going to switch from roundness to diameter equivalent circle. As soon as I set that up, it's automatically classified my globules by those ranges. And finally, we enter the last step. Again, these are all pre-configured, and that means we have some pre-configured graphs and histograms. We also have pre-configured outputs in terms of individual measurements, statistical information, class stats. We also have pre-configured reports. All of this can be fully customized, but again, we want to give you a head start. That way you're not creating graphs and measurement outputs and reports from scratch every time you want to perform analysis. Now, this step here, we're done. We've added everything that we need. We've walked through the protocol. Now we want to set up a few automation steps because, again, the goal is not to walk through this every time. We want to click a button and just have it run through 1,000 images and output the data. So I can have the protocol automatically create a report when I run a batch. I can also have the protocol export results into an Excel spreadsheet. So I have those checked, and now I can save a version of this protocol with all of the changes made. So in step number 4, now we are just going to run the automation. So we'll do this based on what we just set up in the protocol. And I will show you a few different options for automating this workflow. So now my fat globule analysis lives in my protocol library. I can access this at any point. But what I'm going to do is uncheck this interactive button at the bottom. And you'll see that the button changes from that next step guided process and gives me an option to either run selected on a single image, or if I click on run batch, I can open up a folder. I can either batch process the entire folder, or I can select images from within that folder. So now it's following every step that we set up in the previous video. I've told the protocol to automatically give me a report. And so here we can see this is one of our reports. If we're looking at multiple images, it includes some additional graphs and charts. And then it has automatically exported data into an Excel spreadsheet. So I want to look at the other 3 case studies, starting with the seed distribution analysis. But again, keep in mind, for each and every one of these applications, we are going to follow those same 4 steps. So we don't need to learn any new tools. We just have to do exactly what we did with the globule analysis. So with the seed distribution, what are we looking for? Well, first, obviously, we need to identify the seeds. If we're interested in a count or any sort of statistical information, we need to be able to segment the seeds. And you can see here that the seeds, they all kind of share similar color, texture, and they're all touching. So this is something where AI deep learning does a fantastic job of separating objects. So with this image, I've actually just used the general objects pre-trained model. I didn't have to do any additional training, although if I did, it would have been a very quick process. But this is really showing that that one pre-trained model it can be applied to just about any image, and it does a fantastic job. So now that we have segmented the seeds, maybe we want to classify these. And what we did in the globule demonstration is we classified them into bins based on a single measurement type. However, with these seeds, you can see that they're not. I can't use just a single color measurement. I can't use any sort of circularity, diameter. There's just not enough difference between these seeds. So we do have another classification option called learning classification, where I can combine multiple different measurement types together. I can just click on a few reference objects within these classes, and it will automatically assign weights to the different measurements, and it will go ahead and classify based on what I've referenced. So again, just a more robust classification if you can't go off of a single measurement type. Population counts and percentages. This is already set up in the particle size protocol, so no need to do anything. I'm just going to have this pie chart already configured after I classify. And maybe I'm interested in some LAB color values. I want to look at statistical information per class, and I want to look at the L, the A, and the B means. So again, I just have to go in and add these measurement types at the end of the protocol. Now it's going to output this every time I run the automated version. This workflow took me about 2 minutes to set up. Again, I didn't have to train a model. Really, I just had to configure my classification, and I had to set up additional LAB measurements to be output. In the next case study, we're going to look at serial pieces, so serial morphology and structure. And with this, again, we want to find the individual serial pieces. I did this without having to do any additional training to the pre-trained model. But now maybe we need something a little bit more robust when it comes to selecting or ignoring certain pieces. So you can see in the sample image, I have a couple broken or partial serial pieces. Maybe I want to get rid of things that are smaller than a certain area threshold. But I can also combine other measurements as well. So I'm looking at a scatter plot where I can combine area as well as circularity. If I have known ranges or I just need to move this around to get rid of things, that's available as well. So now it's going to ignore all of these partial pieces based on the range. And I can classify like I did in the case study 2 with the seeds. But again, ignoring those partial pieces. So in this case, I'll go off of just a region caliper, a mean, and I've set ranges and 3 different classes. And so now I can do a comparison of diameter, any sort of measurement type. Again, individual measurements, overall statistics. Even if you are batch processing 100 images, I can look at every single object that was counted in those 100 images and get statistical information, all the way down to I can look at the measurements for each serial piece that was counted. Again, similar to the last example, this took me about 3 minutes. I just had a few additional things I had to configure. But this allows me to move from one application to another without having to spend a tremendous amount of time. In the final case study, we are going to be looking at chocolate drizzled pretzels. And you'll notice in this sample, I've got 2 different types of chocolate. So what I want to do is I want to find each individual pretzel first. And I did a little bit of training. The pre-trained model was pretty accurate, but it was kind of splitting some pretzels in half, but the training took about a minute. I think I just labeled one additional pretzel as reference. And then we want to segment the peanut butter chocolate and the dark chocolate because we want to get some sort of ratio or percent area measurement per pretzel. And this could be because I'm doing R&D, or if I'm doing quality control or testing out a new piece of equipment, but I know I have tolerances and these pretzels need to be coated with certain percentages or ratios. What I didn't go through is we've also included additional architectures for AI deep learning. So what we've gone through already is all based on the cell pose architecture, where it does a really good job of finding individual objects. For any sort of area-based measurement, we have an architecture called UNet, where I can actually, with a single model, I can create multiple different classes. So there's a lot of different ways to tackle the analysis, but the point I'm trying to make is that we have 3 different architectures within the software, which also makes us extremely unique. A lot of software includes one or the other, but we have decided to combine multiple different architectures just to be able to tackle any application. So I'm setting up a UNet model with 2 classes, one to find the peanut butter and one to find the dark chocolate coating. So now I've already segmented the pretzels into individual objects, and now I can run the UNet 2-class model within each of the pretzels. And from there, I'm just reporting the percent area measurement per pretzel. So we could use what we call the particle phase, which is sort of an objects within objects. I want to find an object first, then I want to find other things, phases or objects within those parent structures. So now I can get a per pretzel output for the coverage in terms of peanut butter versus dark chocolate, as well as a ratio. And then I can output that as a graph as well. Now, this workflow took about 8 minutes, and it was not structuring the protocol. It was simply training that UNet model to find the 2 different types of chocolate. But again, it's quite a complex analysis process, and we can still keep that within an extremely reasonable amount of time. So that is it for me. I've tried to demonstrate a diverse range of applications, but again, it's all following the same process. So thank you, everybody. If you do have any questions, I think now is Q&A time. I actually kept my portion within my 30-minute allotted amount of time, which is, I think, a first for me.
All right, we'll give a couple minutes for anyone that might want to send in some questions. And just to follow up quickly, thanks Tom. We have the special offer for those attendees today. We'll also send it out, letting you know that we have the combination packages that were highlighted during the presentation today. We offer the promotion to those that attended. All right, appreciate all of you attending. Thank you very much, and keep your eyes open for our next webinar. Thank you very much for attending.